I'd like to do one last question before we turn to the question and answer session. And that's really about your view of the future for this field. It's been evolving quickly, but where are things going from your side of things, and is it going to get easier or harder?
Well, and I think that as we look to the future of where this is going, the idea that we are testing these things one at a time is where we are now, although we're already starting to move out of that mode. And so if you consider that for a patient with lung cancer, there will be an EGFR test and a KRAS test and probably an ALK test all done separately, that is a slow and expensive way to do things that uses a lot of tissue. And increasingly, we're moving into getting more data at a time. And so this is often called multiplex testing. It's more cost-effective per analyte, but the overall cost is much larger because you're looking at many more analytes. But it uses about the same amount of tissue as doing just a couple of tests. So there's the economy of money, but there's also the economy of tissue to take into account here. And where this is really going next, and there are some labs that have already adopted this, is what's called massively parallel testing, or in some cases referred to as next-generation sequencing. And this is much more efficient data collection, but it is quite expensive. And it uses about the same amount of tissue as the multiplex testing. But the real question here is how to use that information. So in next-generation sequencing, we simultaneously test for hundreds and hundreds of things at the same time. This, I think, really is going to drive pushing oncology towards characterizing tumors just as much by those molecular alterations as, wow, this looks like a squamous cell carcinoma, or this looks like an adenocarcinoma, these boxes that as pathologists we put things in based on how a tumor looks. And I think that that might actually break down some of these barriers that we have with some of these boxes that says, this is how the tumor looks, therefore I'm calling it a blank, that will be combined with, this is a tumor that looks like this, but has these other alterations. I think that one of the major goals of this is that it will ultimately bring about a different sort of time for having multiple answers at once. Even though the one test is likely to take a lot longer, it will mean a lot less sequential testing. But there's a caveat to this, like there is to anything, and that is that even with simultaneous testing for hundreds of targets, there's always going to be the newest thing on the block that may or may not be part of that target list. And we always need to keep our eye on what the next interesting development is, because not long ago, EGFR mutations were nothing more than an interesting development, and we have gone on to see how they've really revolutionized how we think about characterizing and treating lung cancer. To me, this really underscores the importance of all of the work that goes on long before things get to the point of standard of care, which is the translational research, the participation in clinical trials, the basic research that gets us there. And to put in my own little plug here, I think all of this requires federal and congressional support for a national research budget, and I think that's a big part of how we are where we are today in cancer research and cancer care.
It's interesting with the thought that we might evolve from having people defining themselves as lung cancer or breast cancer experts to having treatment sorted out as HER2-driven cancer, EGFR-driven cancer, and that might be across different tumor types.
Yeah, and I really do think that there is going to be an increasing drive towards that concept.
You've talked about how quickly this is evolving, and we've seen EGFR go from interesting to mandatory, essentially, and then ALK has become more widely available, and now we've started to talk about ROS. ROS1 mutations or rearrangements are potentially very important, but not widely available. And so, as a general question, how do you see us integrating new molecular markers into a broader population when those markers might only be available at one or two centers at a time right now?
I think that that's really important, because I think that especially when we think about all of the research that happens behind the scenes in the clinical translational world, there's always a new and interesting test that somebody is working on. And the question is, when it becomes really interesting, how does that become something that people get access to and becomes part of the routine thinking? there's lots of issues when we think about the newest, neatest thing on the block. First and foremost is that often these are considered research tests, and there are a number of logistical and ethical and financial limitations for something that is technically a research test. And in a lot of cases, that's something that can only be done under the context of clinical trials with consent for patients that have been seen at a specific institution. Obviously, in that setting, that's not something that's covered by insurance. Even when a test is no longer research, it may only exist at a couple of labs. And that's really part of the adoption curve, where something has gone from interesting to important, and that transition usually happens at a small number of institutions. And as that development gets more and more accepted, there becomes a need to create the access so that it can become more widespread. So there's no question that access to new testing can be very challenging, and it's a very rapidly growing field. And what I can say about this is that even though new tests always start off at a limited number of labs, that if they truly are really important, they do grow in implementation and access. I think we've seen an interesting phenomenon in this world, which is that in a lot of cases, patients know about this new testing before the physician. And in a lot of cases, patients will say, this is an interesting test and I want tested for it, and if my insurance doesn't cover it, that's okay with me because this is what I want and this is too important. I think that this has to be recognized as a very important part of how we think about getting testing done, but I think it also has to be tempered with doing an intelligent approach to utilization. And so I think your example of ROS1 is a great example. We are one of a very limited number of labs in the country that currently offers this testing, but we work hard to educate people sending us this test to know that they should only be sending us this test after they've looked at all of the other available tests, because these are changes that normally don't happen in the same specimen together, and it doesn't make sense for us to use the access to this test for cases in which it's very unlikely to see a positive result. But I think that it's also really important to think about how getting that kind of testing is used in the long run. And I think in terms of ROS1 testing, we're seeing an interesting phenomenon here, which is that we are seeing a lot of cases in which people are bypassing the clinical trial system. So, for example, I have had multiple conversations with oncologists when we've had positive ROS1 tests, where the referring doctor will say that they're going to try to get off-label approval for the drug. And this really is a yin and yang issue, because for an individual patient, this means a much easier logistical course of care. There's probably not travel involved. It's easier logistically. But what it also means is that we are not involving the rare events in the systematic science. And that is ultimately how we're going to get these tests to be part of widespread access. So if people don't enroll in the clinical trials, we will never get the data that allows us to justify making this a widespread test. And so I think that's a balancing act that's very hard to do, and it's obviously very hard for individual patients to say, oh, well, I can get access to this drug in a convenient way, or I can get access to this drug in an inconvenient way. And I think that those are very difficult issues we have to think about in terms of both individuals but also the science as a whole. But since we already discussed some of the newer technology, I do think some of the newer technology may supplant some of these things. And as we have wider scale testing that is next generation sequencing, we may get some of these answers by matter of course.
I think it's interesting how you'd mentioned that patients are potentially, if not often, going to know more than their doctors about these specific things. And we are seeing that particularly with rarer cancers or rarer subtypes, where patients who are ROS1 positive are finding each other and keeping up with each other and may know more than any one doctor, even the experts who are treating one or two or three patients at their site. But these patients are often communicating with each other so that they are more in touch with what's going on than anybody else. And the hope, I guess, would be that enough of these patients would self-aggregate to say, here we are with a couple of dozen people with a rare mutation ready to sign up together for a phase 2 trial that some institution or pharma company may have. hopefully that could accelerate the rate of clinical research. But I agree that it's a big change right now, and it's certainly disruptive to the system as we have it now. All right. Well, with that, it's the end of the hour, and I really want to thank you, Dara, for your great presentation on a very important topic and a lot of interesting questions that are the kind that come up in our online communities all the time, and I know in our clinic discussions as well. So thank you so much for that. I also do want to thank the Longevity Foundation in partnership with Global Resource for Advancing Cancer Education, or GRACE, in making this webinar series and this particular webinar possible. So thanks, Dara, and have a good night.
Thank you so much. I'm really pleased to have this opportunity.
Thanks for listening. If you like and learn from our GRACEcast, you can subscribe on iTunes by just searching for the term Cancer Grace, find podcasts in the subject you want, pick a format of audio or video, and then just click subscribe. It's that easy. And for those of you who don't want to miss any of our programs, there's even a feed for all subjects. You can also find us on YouTube at Grace for Cancer Info, and that's the number 4 in one word, Grace for Cancer Info. Finally, if you haven't been there yet, please check out our GRACE website at www.cancergrace.org. And don't forget that donate button in the upper right. Our content, which helps tens of thousands of cancer patients around the world every month, is made possible by your support.